Dear students or viewers, in this video we'll deal with the exercises of chapter number 5 NCERT Science, that is the fundamental unit of life. The fundamental unit of life is cell, that is the structural and functional unit of life. So let's discuss 10 questions are given at the back of the chapter. So number first is make a comparison and write down the ways in which plant cell is different from animal cells. So I've already written the answers because it will be very a long video if I will write it in front of the video. So I have already written the portion that need to be written, and then I will explain it. You can see answer number 1. I have made a table to differentiate animal and plant cell, and some basic differences I have written. Number first is animal cells are generally small in size while plant cells are larger than the animal cells. In animal cell wall is absent, that is present outside the plasma membrane in the plant cell. So additional wall is present in plant cell for support. Next is plastids, chloroplast, leucoplast, chromoplast, all that are present in plant cell, but there are no plastid in animal cell except an organism that is called euglena because it contains chlorophyll. Vacuoles, there are many contractile vacuoles which are many in number, temporary and small vacuoles are there in animal cell, while in plant cell you have a single large vacuole which is permanent and filled with cell sap. Golgi apparatus is single and highly complex structure in animal cell, while in plant cell it is small units of Golgi complex are there, and there are many units and they are called as dicytosomes as compared to Golgi apparatus. Centrosome and centrioles, which are important for cell division, is present only in animal cell while it is absent in plant cell. You can see all these differences clearly from the diagram of animal and plant cells. Next is prokaryotic, right? You have to differentiate. Question number 2 is how is prokaryotic cell different from eukaryotic cells? Again, I have made a table and written prokaryotic cells are primitive cells, that was bacteria generally, and eukaryotic cells are the fully developed modern cells. So prokaryotic cells are generally small in size, that is 1 to 10 millimeter in size, and eukaryotic cells are generally larger in size. In prokaryotic cell, a well-defined nucleus is absent. Only a single chromosome lies in the center of the cell, while in eukaryotic cells, nucleus is present. Nucleolus again is absent in prokaryotic cell because nucleus is absent. So in prokaryotic cell, both are absent, while in eukaryotic cell, a fully developed nucleus with nucleolus, nuclear membrane is present. Here, only a single chromosome you will see in the nucleus, that is called as nucleoid, and more than one chromosome are present in eukaryotic cell. Membrane bounded cell organelles are absent in prokaryotic cell while they are present in eukaryotic cell. So I hope some diagram may be given in your book of prokaryotic cells. So you can see here this is a prokaryotic cell with only single chromosome lying in the center, no cell organelles there, and it is very small. It is a microscopic structure. You have been given a zoomed structure of prokaryotic cell, but this is a prokaryotic cell, and a fully developed animal cell has been given in your book in figure 5.5. Next is question number 3 is what would happen if plasma membrane ruptures or breaks down. So let's see if what happens. Plasma membrane is present in both animal and plant cells and acts as a boundary for the cytoplasm, or the boundary line for the cytoplasm of the cell, or binds the cell, gives it protection, mechanical and strength and rigidity. It is semi-permeable in nature, so, and it also maintains the constant composition of the cell inside, that is also known as homeostasis. Homeostasis is the condition of the constant composition of the cell. So constant internal composition of the cell will obviously be lost if cell membrane will not be there, and there will be no protoplasmic functions. It will not be able to perform its basic functions, and the cell will get instantly killed. So if there will be no plasma membrane, there will be no basic functions performed by the cell and will gradually perish. Question number 4 is what would happen to the life of a cell if there is no Golgi apparatus. Golgi apparatus is related with secretion. If there is no Golgi apparatus. So there will be no intracellular digestion there, no exocytosis will be there, no formation of new plasma membrane will be there, because all these are the functions of Golgi apparatus. Next is, which cell organelle is known as powerhouse of the cell and why? The powerhouse is known as, is given the term to the mitochondria, which is the site of cellular respiration. Here the cell respires utilizing oxygen from the atmosphere and releases energy in the form of ATP, that is energy currency of the cell, and inosine triphosphate. And as we require energy, the energy is taken from the ATP inside the cell and utilized in various functions like photosynthesis, protein synthesis, etc. And later it is converted to ADP, and on getting again the energy from respiration, ADP is converted to ATP. Next is, where do lipids and proteins constitute the cell membrane get synthesized? So proteins are synthesized on the ribosomes of rough endoplasmic reticulum. You have seen this type of tubular structure and plasmic reticulum. On this, the small ribosomes are present. These are protein factories of the cell. So rough endoplasmic reticulum helps in the protein synthesis, and lipids are synthesized over smooth endoplasmic reticulum, that is endoplasmic reticulum that is devoid of ribosomes. You see, this part is devoid of ribosomes, so this is termed as smooth endoplasmic reticulum, and the one which have ribosomes on it are called as rough endoplasmic reticulum. Question number 7 is, how does an amoeba obtain its food? So you can see here, amoeba is just like an irregular shape organism, unicellular organism, and these are called as pseudopodia. As soon as it sees the food, it develops, the pseudopodia elongates and engulfs the food by endocytosis. So it engulfs the process by endocytosis, also called as phagocytosis, through phagosome, which combine with the lysosome, which helps in the digestion, intracellular digestion, and forms the food vacuole. So this vacuole is called phagosome, which combine with the lysosome inside the cell, and together forms a food vacuole is formed. This food vacuole is formed, and it engulfs the food particle inside the cell, where the food is digested and the undigested or unwanted food is thrown out of the cell. Question number 8 is, what is osmosis? Let me explain you with the help of a diagram. Osmosis, you can see here, let me draw a simple diagram here. Suppose this is a semi-permeable membrane, and here you have, these are some solute molecules, and here these are solvent molecules. Small molecules are solvent molecules, water suppose it is, and here you have again solute molecules and water molecules. Now you can see that here water molecules are more, while here water molecules are less. So the movement of molecules, solvent molecules, from its higher to its lower concentration across the semi-permeable membrane is termed as osmosis. So osmosis refers to the process, especially to the movement of solvent molecules from its higher to its lower concentration across a semi-permeable membrane. Question number 9 is a diagrammatic question. I have drawn already the diagrams. In this you have been given 4 peeled potato halves, and they are kept in A, B, C, D containers. You can see I have drawn 4 containers, and in this I have kept the peeled potato with the central hollow part. These are 4 potatoes. Okay, in A cup it is kept empty. There is nothing inside it, and outside there is water in all the 4. Now in B part, in B tumbler, I have kept 1 tablespoon sugar inside the cavity. In third one, I have kept 1 tablespoon salt inside the cavity, and in D I have kept 1 tablespoon sugar plus the potato is boiled in this case. Now you have to answer the following questions. Keep these for 2 hours and observe the potato cups and answer the following questions. Explain why water gathers inside the hollowed portion of B and C. Now as you see that water collects inside, it is saying that water collects inside this hollow portion in B and C. Why? Because of endoosmosis. How? As you might have read that osmosis is of 2 types, endo and exosmosis, and endoosmosis takes place when the concentration of the solution is more inside as compared to that of the outside. So because here it is sugar, and here it is salt. So the concentration of the solution inside the cup made in the potato is more as compared to the outside. Therefore endoosmosis takes place and water moves inside the potato through the membrane or through the cells of the potato. It moves inside by the process of endoosmosis, and the water level inside the cavity increases because of the movement of water molecules from outside the tumbler inside the potato cavity. That is because of endoosmosis, and that is because in turn because of the higher concentration of sugar and salt solution, which is due to higher concentration of sugar and salt solution inside the potato cavity as compared to the outside, as compared to outside the potato. Okay, so this is basically because of endoosmosis. Next is question, why potato A is necessary for the experiment. You can see in potato A you will see no water inside it. Why? Because this acts as a control for the experiment. You have learned what is a control experiment. Every experiment has its control, and it shows that if there is only water, then alone the potato cavity or potato cells cannot induce the movement of water inside the potato cavity. For that you need a concentrated solution. Either a more concentrated solution or a less concentrated solution is required to show the osmosis. But as far as, so in control experiment you have to basically remove whatever process you are studying, and the thing that is responsible for that process is removed from the control experiment. मतलब जिस चीज़ के बारे में आप पढ़ रहे हैं, जिस फिनोमिनन के बारे में आप पढ़ रहे हैं, जैसे इसमें हम ऑस्मोसिस के बारे में पढ़ रहे हैं, और ऑस्मोसिस के लिए हमको एक कॉन्सेंट्रेटेड सोल्यूशन या लेस कॉन्सेंट्रेटेड सोल्यूशन चाहिए जो यहाँ पे नहीं है. तो बिकॉज ऑफ दैट सोल्यूशन ओनली, उसी सोल्यूशन की वजह से हम एंडोऑस्मोसिस या एक्सोऑस्मोसिस शो कर सकते हैं. तो जब वो सोल्यूशन ही नहीं है तो खाली पोटैटो सेल्स से आप ऑस्मोसिस शो नहीं कर पाएंगे. दैट इज सेमी परमेबल मेम्ब्रेन से हम ऑस्मोसिस शो नहीं कर सकते हैं. जो यहाँ पे प्रेजेंट है और यहाँ पे सॉल्यूशन प्रेजेंट नहीं है या कॉन्सेंट्रेटेड सोल्यूशन प्रेजेंट नहीं है, तो दैट एक्ट्स एज अ कंट्रोल फॉर द एक्सपेरिमेंट. हर एक्सपेरिमेंट का अपना एक कंट्रोल होता है, और ज्यादातर केसेस में आपको दे कैन आस्क यू टू ड्रॉ ऑल्सो, वो आपको ड्रॉ करने के लिए भी कह सकते हैं, और किसी किसी केस में ऑलरेडी ड्रॉन होता है. आपको आइडेंटिफाई करना है कि कौन सा कंट्रोल एक्सपेरिमेंट है. तो यहाँ पे ए जो है वो एक कंट्रोल की तरह एक्ट करता है, तो एज अ कंट्रोल एक्सपेरिमेंट. नंबर थर्ड इज एक्सप्लेन वाई water does not gather in the hollowed out portion A and D. तो A because it is a control experiment, यहाँ concentrated solution है ही नहीं, तो यहाँ पे तो होगा ही नहीं. और D के केस में जो potato है, it is already boiled and boiling kills the cells of the potato and therefore no endoosmosis takes place. So in D the potato cells are killed due to boiling, and in 1 it is a control experiment. A is a control setup. So therefore there is no endoosmosis. Which type of cell division takes place for growth and repair of the body? That is mitosis, and for the formation of gametes meiosis takes place. So for growth and repair the division that takes place is mitosis, and for the formation of gametes the meiosis division takes place. So these are the questions of chapter number 5, that is the biology's first chapter, and next in the next video I will deal with the physics first chapter back exercises given at the back of the chapter.